Hi, I'm Leslie. I'm from University of Florida in Kylie Graham's lab, and today I'm presenting my recent paper, Equitable Machine Learning Counteracts Ancestral Bias in Cancer Genomics. So we know that all modern humans migrated out of Africa, and as populations migrated, they underwent these unique genetic bottlenecking events that drastically reduced the variation in their genomes. Then as they settled across the globe, they started to regain variation and gain variants that were unique to help them survive in their current environments. And so this results in populations across the globe having unique levels of variation. And one of the biggest, um, sorry, I'm going to get rid of this. Uh, One of the biggest studies looking at this and trying to measure this variation across the globe is the 1000 Genomes Project. And so they sampled 26 populations around the globe, shown on the left here. And on the right here, we have those 26 populations on the y-axis. And the x-axis is showing us variants in each of those populations that's rare across the globe, but common within a population. So again, just looking at how different populations have variants that are unique to them, especially we see high levels of variance in African populations, which is this group of yellow populations down here. We also see high variation in Peruvian, just showing that across the globe, we have different variants. And so in when we think about what kind of effects this can have in disease and predicting with models, we have those genome-wide association studies that Nick Kell talked about. And we see that we have a really strong overrepresentation of European individuals. And so when we try to predict traits based off these individuals, we see that we're able to do it really well in the data that we have. So on if we use European data, we can predict traits really well in European individuals. But the further that we get genomically, from that training data, the worse our models are doing. And so we see a similar effect in gene expression. This is the ancestral makeup of the Cancer Genome Atlas data. And so again, we have an overrepresentation of European individuals. And we see similar ancestral effects in expression data. So this plot is showing the average expression of 4 genes across 3 ancestries. So we have green is East Asian, blue is European, and yellow is African. And this is plotting average expression after we've regressed out, or they've regressed out the subtype information and batch effects. And so normally we would expect all of these expression genes to just be on the origin of the graph, but instead we do see differences in baseline expression based off ancestry. And the problem is that we're trying to model really complex population structure and different variants in our models when we're really only giving our models a really small subset of information to actually work from. And of course, the best thing to do is to try to increase representation of these diverse ancestries in our disease databases. But this is, of course, really time and resource intensive. So in the meantime, we're looking at ways at how we can try to utilize available information that's already out there and incorporate that into our models to improve predictions. And so that's basically what our project is trying to do. And we're trying to do that in gene expression data. And we're trying to utilize the publicly available population genomics information from Nomad and 1000 Genomes. So that brings me to our method. And this is my advisor, Dr. Graham, and our 2 collaborators on the project, Dr. Cahill and Dr. Lee. So the first thing that we wanted to do in this is to basically look and see at baseline, if I train a simple regression model in European population, and I train another regression model in an African population, what do those signatures look like in each of those populations? Do they overlap? Do they interact? So we went ahead and did that. And so this is showing the overlap of a model trained on African and Europeans, and it's projected into a functional interaction network. And we see that at baseline, we don't have much connection between the 2 gene signatures of the ancestries. But if we project the entire network, so all genes used to predict in Africans and all genes used to predict in Europeans, in red and blue here, we see that those nodes are actually really central to that network. So we have kind of a shared subnetwork. And we can actually get from a gene that's relevant to disease in Europeans, in this case, and to a gene that's relevant to Africans within about 2 hops in this shared network. So it's kind of giving us a way to kind of navigate a network and get to genes relevant to each ancestry. But then we have to actually try to identify genes that are relevant to an ancestry. So to do that, we have created this metric called enhanced allele frequency. And this is calculated from a publicly available database. So here we use Nomad, which gives us 8 populations. And we calculate enhanced allele frequency for every population, for every variant. So for example, given one variant, I'm going to take the allele frequency in one population and subtract the average of the allele frequency in the other populations. And so what that baseline tells me is this variant found more commonly in this ancestry? Is it found less commonly? And it gives us a way to kind of integrate that information from other populations that might not be represented in my training data. And so this plot on the right here is showing the distribution after we've calculated EAF in Nomad. And so we're seeing this, the top one is just all of our enhanced allele frequencies, and then the bottom one is showing in the frequencies in cancer-specific genes. And so this is just showing that we've calculated EAF from a healthy tissues. It's not unique to cancer, even though we're applying it to cancer in this case. And we also have this threshold here at 0.2. And so in our method, which I'll talk about on the next slide, we're targeting variants above that threshold because we hypothesize that genes that have variants that are different between ancestries are more likely to be underrepresented and not caught in single ancestry models. And that brings me to the method. So we're integrating both the idea of that interaction network and our EAFs. So our model will take in RNA-seq data or any gene expression data. And we do an initial logistic regression run with a lasso penalty to get a really small subset of genes relevant to the disease. We then use those genes as start nodes within our network propagation. So we put those onto the network and we take up to 2 neighbors from those initial genes that then get passed on to our enhanced allele frequency filtering here. So then in our model, the enhanced allele frequency is done kind of for every ancestry. So for a gene to be associated with an ancestry, it has to have a variant that is uniquely enhanced in that ancestry and above that threshold of 0.2. And so here in this step, we end up basically with every ancestry having kind of a list of genes that qualify and represent that ancestry. If that list is longer than 30 genes for every ancestry, we're going to go ahead and take the top 30 for each ancestry, top 30 most variable genes from the training data, just because we want to keep our signature fairly small. So that gives us about 150 genes. We put the genes for every ancestry into one gene list. We add back in those start nodes, and then we rerun the regression model using a ridge penalty to force inclusion of every gene into the final model signature. And we're comparing against a benchmark, which the layout is the same, except it's not getting any enhanced allele frequency filtering or network propagation. So again, we're starting with the RNA-seq data, do our initial logistic regression with our lasso penalty. Then it's going to get a random subset of the most variable genes in the data set. And this is going to be the same size as the phylophore signature. We rerun the logistic regression with the ridge penalty to get our final comparable benchmark signature. So for model training, we're going to use TCGA, the Cancer Genome Atlas data. And we picked the 3 most diverse cancers. Of course, they're not that diverse, but so we have breast cancer, uterine cancer, and thyroid cancer. And in order to train our models, we basically wanted to be able to compare performance both within a single ancestry and across ancestries. So we end up breaking up for every disease individually. We end up breaking up our samples into these like smaller batches. And we're going to break up our batches based off the ancestry that has the smallest sample size. So here, our admixed population is the smallest. So we're going to break up our other 3 ancestry populations based off this sample size. So East Asian gets 1, Africans get split to 2, and European ends up with 17 batches. So I think this is actually the numbers for the breast cancer run. So in total, we have 21 batches here. For every batch, that batch is going to be trained both on phylophore and on benchmark. Then that trained model is going to be applied to the other 20 batches that are for that disease. So at the end of training one single batch, we're going to have 20 performance metrics, and then one signature for phyloframe, which is our method, and one signature for benchmark. So in total, across all of our diseases and models, we're going to have 1,401 models trained on our method for phyloframe and 1,401 on the benchmark. And just a note that you don't, the ideal point of our method is that you don't actually need to know your sample's ancestry when you run it. This is just for us to kind of benchmark and look at performance across a known sample. And then this is an example output just from one run of phyloframe. So both the benchmark and phyloframe are obviously going to give you the gene list that they use to make the prediction. Then for phyloframe, we can actually go back and we can look at why a specific gene was included in the model. And so this heat map is showing the genes as the columns, and on the rows we have our nomad ancestries. And if the box is colored light gray, it means that gene was not included for that ancestry. If it's dark gray, the gene was included for that ancestry as well as another ancestry. And if it's color, it means that gene was included specifically for that ancestry. So just showing that we can actually go back and actually look at why genes were chosen. And so, of course, we'll have that for phyloframe. And then we'll have 20 sets of metrics to look at for every run. And so this is looking at those metrics. And here we are coloring the dots by the training data. So these, for example, here are all trained on European. Blue is trained on African, etc. So this is our breast cancer run. We're doing a binary subtype prediction, and we see significant improvement, particularly in the MCC and recall. And also, interestingly, we see this more in the breast cancer than in the other two, but we see a majority of the improvement in our model predictions is actually done on the models that are trained on Europeans. So those models that actually are trained on more diverse ancestries, such as African, actually don't benefit as much as the ones trained only on European. Uterine cancer was also a binary subtype prediction. And again, we see improvement with our model, primarily, again, in MCC and AUC, not as much in recall and precision. And then this in thyroid, we actually did a metastases prediction. So this prediction is pretty tough and sometimes I think has a lot more labeling issues than subtype, but we don't see really any improvement with phyloframe in this prediction. The next thing we wanted to do was just evaluate the signatures that our model is using to actually make the prediction. So this is looking at the breast cancer model, and we're looking at the genes that were used to make the prediction. So up here in A, we have the overlap of all the signatures for both our model, phyloframe, and our benchmark comparison. And here we see that phyloframe is doing a much better job at picking the same genes in every model. So it's showing a lot more signature consistency across models, which makes us more confident that we're actually picking up a biological signal. We also see this is, again, with cosmic cancer genes, so the known cancer genes here. We again see that it's picking the same, more consistently picking the same cancer genes throughout the models when compared to the benchmark down here that's not as consistent. And then this heat map is kind of a larger version of B down here, where we're looking at any known cancer gene that was picked up in benchmark or phyloframe. And again, we see phyloframe has majority of the genes, the known cancer genes it picks are actually picked throughout models and across multiple models, versus benchmark is kind of picking up sporadically different cancer genes, but it's not doing it in a consistent way. We also did a global validation set. So here we looked for any transcriptomic data set that had ancestry and subtype information. And so we ended up with 14. We classified them to the 5 continental ancestries. And here we're looking at basically every plot point is going to be one individual. and we're looking at how many models correctly predicted that individual. So if I have 16 models and 16 models, and 8 of those predicted an individual correctly, my dot will be at 50 here. And so again, across ancestries, we see that our model is improving performance, particularly in African, East Asian, and European. And then the last thing we did is try to look at how our model performance is correlating with phylogenetic distance. So here we sorted our samples into non-African, East African, and West African. And this over on the left is showing the phylogeny for those populations. And we see that for again, this is showing the percentage of models that correctly predicted a sample and for the European models. And so we see that for the samples closest to the training data, our models do the best. So we have East Asian and European kind of grouped together in non-African, and we have the best model performance. And then as we kind of follow the phylogeny away from Europeans, we see our performance get slightly worse. And that's it.
All right, thanks, Leslie, for that really great talk. As the previous moderator mentioned, you can put any questions you might have in the Q&A box. If you look at the bottom kind of bar, it just says Q&A, and you can type in your question there. I guess I had a question. I was kind of wondering, so these are all, I assume, bulk transcriptomic data sets that you're working with, or any of these single cell? Yeah, I was wondering if, like, what your thoughts might be on whether or not you could use deconvolution to see like specific cell type drivers and kind of link those to ancestries with these current data sets, or if you're thinking about extending this to like existing single cell data sets.
I hadn't thought about kind of looking at cell type with this data set. I think that would be very cool. We have talked about applying it to single cell data sets, and I think it would it would still work. We've actually have talked with a collaborator to try and do that. But yeah, no, that's a great point.
Other moderator, yeah, go ahead.
Yeah, I had a question too, actually. So my sense is that you're capturing kind of private variants to these populations that are near genes, is that right? Are they actually functionally involved with the gene, or are these just kind of you're just looking for enrichment of variants in general near genes that matter, if that makes sense?
We're looking at variants on genes, so only on the exome. Yeah, sorry. Did I answer your question?
Yeah, no, that makes
Yeah.
sense. Yeah.
We wanted to extend it to the whole genome, but of course it's hard to start associating.
Yeah, no, exactly.
'Cause-
And then there's the whole problem of like there's going to be differentiation anyways, right? So then it's like which variants matter is a problem, right? So
yeah.
yeah, exon variants is a good idea though. Yeah, that makes sense.